Hello everyone, a very warm welcome to all of you. The topic for today's discussion is vulva development in C. elegans. This is a very important topic from developmental biology syllabus. So today we'll get to learn how vulva develops in C. elegans. So as we all know, C. elegans are individuals that are hermaphrodites. Okay. They are male in their early developmental phase, and gonad produces sperms, which is stored for later use. Ovaries develop as the nematode grow older, and the eggs roll through the region of sperm storage. They are then fertilized inside the nematode, and then they pass out of the body through the vulva. Now, this easy, simple diagram will help you to understand the process better. See, this VPC, this stands for vulval precursor cell. Okay, vulval precursor cells. This cell here is the anchor cell, and here you can see the gonad. Now, these are the VPCs or vulval precursor cell. Now, this is just elaborated here. You can see the numbers P3 through P8. This is the basement membrane and this is the cuticle. In between, you get the vulval precursor cells. See, the adult vulva comprises of 22 cells, which are progeny of 3 ancestral cells originally located in a row on the undersurface of the developing worm, right? So if we take a note of this, see, so adult vulva comprises of how many? 22 cells, okay? These are progeny of 3 ancestral cells. Ancestral cells. Now, these 3 ancestral cells, they are originally located in a row on the undersurface of the developing worm. What we have here, see, you have the anchor cell. You have the anchor cell here. Now, this anchor cell, remember, this anchor cell is the inducer. This anchor cell is the inducer. What does it do? It induces these cells. You have another set of cell here. Sorry. Okay. This one will adopt primary fate and these two will adopt secondary fate. Now, what is happening is what we are going to study now. As I've already told you, the vulval precursor cells are 6 hypodermal cells. You can see it from here. See? 1, 2, 3, 4, 5, 6. 6 hypodermal cells numbered as P3P through P8P. And these 6 cells form the equivalence group. Okay, these 6 hypodermal cells are forming what? They are forming the equivalence group. The central cell, or this P6P, this one adopts the primary vulval cell fate, while P5P and P7P adopt secondary cell fate. Now take a look at the picture nicely. This one, this P6P here, it becomes primary. The adjacent ones become secondary. Clear? So this is what is happening. The anchor cell is there. This is your anchor cell. This anchor cell induces the hypodermal cells or the vulval precursor cells. Vulva forms during the larval stage from 6 cells called vulval precursor cells, and Lin3, here see, this Lin3, you have heard of EGFs. This Lin3 is homologue of EGF. Okay, the anchor cell secretes Lin3 protein, which is encoded by Lin3 gene. Okay, it forms the concentration gradient, and thus P6P, being closest to it, adopts primary fate. You can see here P6P is closest to the anchor cell. So this comes under the direct effect of the gradient. As I've said, Lin3 is produced by anchor cells, and this is closest. So it gets the maximum effect and it adopts primary fate. These two are little far away, P5P and P7P. So they become the secondary, they adopt secondary fate. They are the lateral vulval cell, and this one is the primary vulval cell. Now see, we should know the cascade of events. What is actually happening? The cascade of event is very important. The anchor cell is producing Lin3, which is forming a gradient. This is directly influencing P6P, but this has to be kept in mind, something called lateral inhibition. This is very important. Lateral inhibition plays a very important role here. Now, who is controlling this lateral inhibition? Lateral inhibition occurs through Lin12. Lateral inhibition is occurring through another protein called Lin12. At the two sides of P6P, you can see P7P and P5P. These two vulval precursor cells are present. Now, these two come under the influence of Lin12. lateral inhibition occurs through Lin-12. This has to be kept in mind. The P6P part here you can't see, I guess. The P6P part is the central vulval lineage. This one. And the lateral vulval lineage is the P5P, P7P. This is the lateral vulval lineage. Right? The receptor for Lin3 is Let23. See, here you get to know, get to learn about a lot of numbers. Lin3, then 12, 23. So please don't get confused. You should remember the name of the ligand and its corresponding receptors also. So the receptor for Lin3 is Let23, receptor tyrosine kinase. The signal is then transferred to the nucleus through RTK-RAS pathway. Clear? Lin31 protein is the target of the kinase cascade. Now what is happening? See, phosphorylation of this protein in the nucleus causes it to lose its inhibitory protein partner and allow it to function as a transcription factor promoting cell fates. Now, the vulva progenitor cells are also subjected to regression signal from hypodermal cells. This regression signal has to be overcome. This regression signal is overcome by positive signals, that is like induce P5P, P6P, P7P to differentiate and prevent unwanted differentiation of adjacent cells. Now, what is happening? This regression signal is actually preventing the adjacent cells or that lateral cells to, they doesn't, in simplest word, the regression signal or the regressive signal doesn't allow the these lateral cells to differentiate, and and this unwanted differentiation is prevented in order to give rise to a correct, proper vulva formation. If all the cells are differentiating in their own way, then correct formation of vulva wouldn't have taken place. So this lateral inhibition is very important. Had there been no lateral inhibition, there would not be proper development of vulval cells. So this is our today's lesson. It's a very short one, but a very important one. This lesson is really very important. You should all know the different proteins and its receptors and which is controlling which one. Which function is being controlled by which protein. If you know this much, it's more than enough. It is not too big. You don't have to remember too much of contents on this. But if you just know this much, you will be able to answer all possible questions. Okay, till then, thank you so much. See you soon.